Hello and welcome. In this video, we're going to run through the gene molecular evolution workflow on the FormBio platform. What I've done is I've just started typing in the name of this workflow in the upper right, which is why it's the only one showing. I'm not going to go over every aspect of this intro page, mainly because I know we have other videos that also explore the important things that are in these tabs, and inherently we'll go over some of this as we walk through this workflow. In the gene molecular evolution workflow, you actually have 4 different options of how you want to run this. In the curated gene family option, what you're going to give the workflow is a multi-FASTA file that has the sequences of those genes that you're interested in. In the curated orthologs workflow, you're actually just going to give a gene name or a gene symbol to the workflow, and it will automatically pull those ortholog sequences from elsewhere and create a multiple sequence alignment and phylogeny from that. You can also just run OrthoFinder. In order to run OrthoFinder, you have to point the workflow to a folder that has all of the gene annotation index files that are created automatically when you build a genome in the FormBio Build Genome Indexes workflow. So you can actually have multiple species and it'll run OrthoFinder on all species that you have in that folder. And lastly, you can create an MSA from a database sequence search. If you select this option, what you're going to do is you can actually give the workflow one sequence, and then it will go to the UniRef site and pull more sequences from their database and create a multiple sequence alignment and phylogeny from that. For this demo, we're going to go ahead and do the curated gene family version. I already have a metadata file. What the metadata file does in this case is it links your samples to a group that you're interested in. In this case, I just pulled the FGF10 gene from NCBI for 7 different birds, and I put those in the groups flighted or flightless. And what I have is a FASTA file that has the sequences in the sample ID column, and the sequence headers are named just as you see them here. So I'm going to select that file for 7 birds, and then we can hit next step. One important thing to remember is if you type in the sample ID and group in here, just remember to hit the save button before you move on if it's not already something that's saved in a folder. Here we can stick to the default parameters. If you want to stray from default, feel free to look at the right side panel for more information. I'm going to run this, but I also have one that's already ready just to show you the output. You get a phylogenetic tree, sequence conservation, sequence difference by group, and some other interesting information. You can see here in the phylogeny that the flighted and flightless birds are different colors. So we have coded that information. So if you're looking for, interesting differences between 2 groups of birds in this case, right? Here's a PCA analysis. And lastly, I just want to get down to the multiple sequence alignment where you can change the color scheme and the visuals on this, and you can scroll across. So all in all, this gives you a lot of information about maybe a gene family that you're interested in and a group that you're interested in. So thank you for watching.